Hi, I'm David Snead, and I'm in the lab of Samara Reck-Peterson and Andre Schlesinger at UC San Diego. Parkinson's disease is a devastating, progressive, neurodegenerative disorder affecting over 10 million people worldwide. Parkinson's patients classically present with a resting tremor and slowed movement, but as the disease progresses, patients can also develop dementia, eventually losing the ability to perform basic activities of daily living. Parkinson's is caused by the death of a particular set of brain cells responsible for controlling movement. But despite decades of research, we still don't really fully understand what causes those cells to die, and we don't know what causes a certain person to develop the disease. There are a few treatments available to alleviate some of the symptoms of the disease, but no available therapeutics alter its progression or do anything to prevent or reverse the cell death that causes the disease. I am interested in understanding at a molecular level what causes Parkinson's disease. The ultimate goal is that a detailed understanding of the precise causes of the disease will then inspire new and better therapeutics targeting the underlying mechanisms of disease. I'm interested in the structural biology of Parkinson's disease. I want to know at the smallest level, down to individual atoms, what the biological molecules that cause Parkinson's disease look like. There's a class of molecules known as proteins that act as molecular machines and molecular scaffolds in the cell. And in many cases, the aberrant behavior of certain proteins can lead to cell toxicity and to disease. By understanding the three-dimensional shape and architecture of important proteins, we can then figure out what these proteins do, how they function, and how they dysfunction, much like how you can tell that a Ferrari has a different function than a pickup truck just by seeing how they look. Importantly, atomic-level protein structures also allow us to figure out how proteins are targeted by small molecule drugs, and they allow us to design and to develop better drugs, altering these proteins to mitigate their toxic behavior that leads to disease. Though many different proteins contribute to Parkinson's disease, one particularly critical protein is known as leucine-rich repeat kinase 2, or LRRK2, or as I'll refer to it, LRRK2. My primary experimental goal is to use a technique known as cryo-electron microscopy to solve the first high-resolution structure of this important protein. LRRK2 is a large, complicated protein containing multiple regions with different functions. Its so-called interaction domains can bind to other proteins, while its enzymatic domains actively modify other proteins. So far, we and others have been able to determine the structures of bits and pieces of different fragments of LRRK2, but we haven't been able to piece them all together to figure out what the entire protein looks like. LRRK2 has a number of different functions within the cell, but our understanding of the molecular mechanisms underlying its function is limited by our lack of high-resolution structural information. Mutations in LRRK2 can directly cause Parkinson's disease, but we don't fully understand how these mutations alter the protein to cause cellular toxicity. Even in patients without LRRK2 mutations, its normal function seems to go awry to contribute to disease. Specifically, overactivation of the LRRK2 kinase domain, one of its enzymatic domains, can be toxic to cells. And in fact, most of the disease-linked mutations effectively increase the kinase activity of the protein. This suggests that inhibition of this domain could be therapeutically beneficial, and a number of LRRK2 kinase inhibitors have been developed and have entered phase 1 clinical trials. Unfortunately, in many cases, drugs can alter proteins in unexpected ways, or can non-specifically inhibit other off-target proteins, which can lead to toxic side effects in patients that ultimately limit the utility of those drugs. We need to understand at the molecular level exactly how drugs bind to and alter their target protein in order to design and to develop better, more specific, and less toxic inhibitors. For all these reasons, a high-resolution structure of LRRK2 has been an important but elusive target. Over the past few years, there's been a so-called cryo-electron microscopy revolution with significant technological advances that only now make this a feasible goal. So far in our lab, we have used cryo-electron microscopy to solve a high-resolution structure of a truncated version of LRRK2 containing the kinase domain and its surrounding domains. And this structure has already revealed some interesting mechanistic insight into LRRK2 function and regulation. For example, our structure suggests that other LRRK2 domains may regulate the activity of the potentially toxic kinase domain by directly physically contacting it and changing its shape. We've also started to identify the specific regions of LRRK2 that mediate its interactions with other proteins. It may ultimately prove therapeutically beneficial to modulate these types of protein-protein interactions, but we will need to understand their structural basis in order to devise ways to alter them. We have found, quite unexpectedly, that though different LRRK2 inhibitors all can inhibit its kinase activity, they actually can have drastically different structural effects on the protein. These structural changes can alter the interactions of LRRK2 with itself and with other proteins in ways that may be detrimental to cellular health. Because these types of unexpected off-target effects can lead to toxicity and to side effects in patients, our structural information allows us to choose which inhibitors seem more promising than others as we try to optimize these drugs and then move them to the clinic. Moving forward, I am working to solve the first high-resolution structure of the entire full-length LRRK2 protein. By then studying how its structure changes in different conditions or upon binding to different proteins or inhibitors, we will better understand how LRRK2 functions, how it binds to and modifies other proteins, and how it is regulated. I also aim to determine how LRRK2 structure is changed by disease-causing mutations in order to determine how these mutations alter its properties to cause disease. Altogether, this work will yield crucial insights into LRRK2 function and dysfunction, and hopefully allow us to develop even better therapeutics for patients with Parkinson's disease.